All right, everybody. It's a few minutes early, which it's very unusual for me to actually start early. I usually start late, but since almost everybody's here, I just have a few announcements to make before we get started. So the first is, and I sent you all an email on this, but the uh, the, the the geniuses, I mean that, you know, literally in the neurophotography, managed to reduce the size of the recording from 10 gigabytes to 400 megabytes. So that's quite an accomplishment. So now what I'm going to do from now on, assuming, so he's going to go, Tony is going to go back and he's going to redo all the recordings, and not redo them, but compress them all. And then I'll put them all on my courses, so you can just access them on my courses. And I'll continue to put the giant files on OneDrive, just in case for some strange reason you want to download those giant files so that you can, you know, watch on your HD television or something like that. So, but anyway, so that's going to be that's a big improvement. So we wanted to keep downloading those gigantic files. And then I'm working on the midterm right now. So as I do that, I'll be able to sort of like put together, while I'm putting together the midterm questions, I'll be able to put together some practice questions. So I know I've been promising that, and I haven't forgotten about it. I'm working on it. So I want to give you guys some practice questions. And keep in mind that the purpose of the practice questions really is to give you an idea of what the questions are going to be like, right? To give you practice with the style of questions. And after I've given you the questions, you've had a chance to look at them, then we can discuss maybe next week a little bit about, you know, what the style is and what things to look for and what things not to look for. But the questions really are, I'm not going to give you this vast set of questions, you know, to help you study for the exam, because good questions are really, really hard to make. And so I'm very, you know, I really am protective of the questions that I've developed that I think are actually good questions. So I'll give you some just to practice with in terms of how to approach the questions. So today you've heard the last from me in terms of content. So from here on in, we've got 3 other lecturers that are going to be giving the rest of the course. And today we have Dan Guitone, who's my neighbor. He's been my neighbor on the 7th floor, you know, for 20 years. He's been my neighbor there for 20 years, and it seems like just yesterday. But in any case, so Dan is a world expert in the eye movement systems, which is a really, really nice examples of neurosystems. And so I think this is a really, now that we've had a little bit sort of introduction to some of this stuff, I think this is a really great opportunity for you to really sort of dig more into the weeds and really go more deeply into the functioning of one system that we actually understand quite well. Okay? So with that, I'll let Dan take over. And they like to ask questions.
No
So
problem.
yeah, so
Sure.
don't hesitate to ask questions if you have them.
Yeah, well, hi everybody. I've been working in the ocular motor system for years. I began
Dan,
life.
can I just say one thing? For the recording, the recording doesn't.
Oh,
You see
all
what I
right.
mean? So just stay
Sure.
close enough that you actually.
Okay.
You don't have to stand right next to it, but you know what
Okay.
I mean.
Sure.
Like
I
what
understand.
Joe was doing was wandering
Right.
over.
Okay.
Just
So when I started off in neuroscience, I was drawn to the ocular motor system because it was a system that had been, you might call it, attacked by, in the positive sense, by engineers. And the very first person to make huge insights into how this system works was a control system engineer who was a professor at Johns Hopkins, and his name is David Robinson, and I will bring up some of his observations as we go through the work. Is that okay, Dave?
I just might have left something down.
Okay. And I should tell you now that the ocular motor system is probably the brain's best understood system, in that you can actually make a wiring diagram, a functional schema of how things work, where you actually put in neurons in the circuit. And you might notice this very old-fashioned equipment down here, which I brought. The reason I did that is because I find it useful if one is going to review a circuit to do it interactively as a group, rather than just flash a few PowerPoint slides, which you look at and are virtually incomprehensible because there's so much on them. So I thought that I'm going to try to have a go at this at the end when we've gone through all the details to see if we can actually together make with all your contributions, draw up the circuit diagram that enables the brain to make a saccadic eye movement from one point to the other. Okay? So now let's start on the actual PowerPoint part of the thing. I'm sure you've been here now for 5 or 10 minutes, so you know you've read this slide. The eye movement system is probably, as I mentioned, the brain's best understood control system. Why? Because the eyes move in the unchanging viscoelastic medium of the orbit. Now, this means that essentially the eyes are moving in their orbits without ever the load changing. This is very different than if you consider limb movement control, which is subjected to varying loads at all times. You have to think of just your arms are always dealing with variable loads. If it only is carrying a bag or wearing your clothes even. The eyes are, throughout your life, the eyes are dealing essentially with the same load, which changes very slowly over decades and decades as you go from being a teenager, say, to an old person. The other reason the eyes is easily understood is that they can easily be measured. After all, they're just a ball rotating in a joint in the orbit. And now modern cameras can record the position of the eye in 3 planes, which is horizontal, vertical, and torsion, with these cameras that can operate at 1 kilohertz. The eye system can also be subdivided into different subsystems, which I'll consider in the next slide. And perhaps foremost is that the neurons that are important for driving and controlling the eye movement systems, they're easily to access. It's a complicated brainstem circuit which is subjected to commands from higher up cortical areas, but the brainstem system we know rather well. There's still a lot of stuff that we don't know, but in terms of understanding the brain, we can say that it's well understood. And that given that we know the brainstem, what the brainstem expects as a command for a movement, then we understand better what the cortex is trying to do as it commands various voluntary actions. In a sense, if you want to understand what what the message is to be, you have to understand what the messenger is trying to say and who he's addressing. So we see this relationship between the cortex and the brainstem. Right? So now let's move on here. So this slide shows you the different systems. I have to speak in this thing, so I can hardly move away to point. I guess I could do it this way, but yeah, okay. So we have basically 2 systems. One is the target acquisition system, target tracking, and the eye stabilization system. So first, I should start on the right and talk about the stabilization. If you don't stabilize your visual axis in space, then what happens is that whenever you move, the image of the world translates on your retina. And the retina is what you might put it in the terms that it doesn't like retinal slip. The retina will give you blurred vision even for tiny relative movements of the visual world relative to itself. So to see clearly, you have to first stabilize your eyes in space whenever you move your head. And since you're moving your head all the time, you need a very good and accurate system which responds at all frequencies, whether it be low, very slow head movements, or very fast like this, or as you're walking and running where your head is subjected to rather high frequencies. All these movements of the head in the external world have to input into the eye movement system to stabilize your eye in space. Make sure your visual axis stays stable in space. So the two systems that do that are the optokinetic system and the vestibular ocular reflex. This system operates at low frequency. If you move your head slowly like this, then the eye goes opposite to the direction of your head and makes sure that your visual axis is stable. And this can operate at very low velocities, for example, and is present in virtually all animals that have a vestibular ocular reflex. For example, in the rabbit, the rabbit if you keep the rabbit's head fixed, the eyes will compensate for motion of the Earth relative to the sky. That's one revolution per 24 hours. So it can respond very slowly. But at higher frequency, it ceases to operate for simple reasons that you know that the delay between the visual signal that impinges on the eye and the output of the retina alone is about 35 milliseconds or so. That puts a severe limit on how vision can stabilize the eyes at high frequency. So there's the vestibular ocular reflex, which takes that job and assures that your visual axis is stable in space as you move your head, for example, as you're walking and running and so forth. And you can just see, if you just move your head like that, if I do that, you're still stable I could still see you clearly, even though I move my head like that. So that's done by the vestibular ocular reflex, whose sensors are in the inner ear, the semicircular canals. And that we're not going to go into because we can't focus on all the systems. The other are the target acquisition systems. There are three of them, essentially. One is called the saccadic system, and this entire lecture will deal with this system, but at least I'll tell you that the other systems exist. So here, the eye faces a fixation point which goes off, and another light flashes in periphery, say, and the eye will generate a rapid movement here, shown the saccade here, to the target, which appeared here instantaneously at 20 degrees. Now, this timing, the delay here, you see, is of the order of 200 milliseconds, even smaller. So it's a very rapid response, and the eye movement is very fast. And like I say, we're going to focus on this system here. But another system, which you all intuitively can understand if you've watched a tennis match or go to a hockey game and watch the puck, is that if the target that the eye is looking, if it starts to move, say to the right, the eye will follow it. We all know that. And this is an example. This is time here. And this is the angle in degrees, and say the target's movement like so. It's moving at constant velocity at 10 degrees per second. There's naturally a latency, a reaction time, which is the order of 200 milliseconds, slightly smaller. And then the pursuit movement starts, but of course it's lagging the target, and there needs to be a catch-up saccade, and then the eye stays on the target. With actually no delay, and so which means that there's a predictive element in being able to follow the target so accurately and keep it on the fovea. So you need a prediction system too, which doesn't simplify things in understanding. The other system is the vergence system, in which the eyes are moving separately. As the target here moves towards your nose, the two eyes are moving in opposite directions. This is called the vergence system. Whoops, I lost track here. Now here, this is the only system in which the eyes are moving in different directions. In the other systems, the eyes are all moving conjugately. We say that means they're moving together. All right. So, and feel free, I should say, to interrupt if you wish, if you have a question. I may not be able to see you so clearly, so let me know. Okay, so this is an example of a saccade. It's a rapid movement which occurs here it's a 20-degree saccade. You see this? Yeah, okay, which occurs here in about 70 milliseconds, but at 20 degrees saccade can also occur in about 40 milliseconds. There's variability between subjects. So if I have an animation here, which has always worked, and I hope it will also work now. No, as always. Try here again. How come this is not working? Sorry, how come this is not working? Dave, I did this. I showed you it was working. Okay, let's try here. Is it? No. We'll try. I guess I'll just give up. We'll see here in just a second. No. I had it working. Odd. No. Okay. If you'll excuse me. So if you can believe me, I showed it to Dave Ragsdale not long ago. Huh? It's true. So anyway, this just shows you that if the eye is looking straight ahead and target appears at 2, flashes at 2, at position 2, the eyes will move very rapidly from 1 to 2 within, say here, 70 milliseconds, often 40, from position 1 to 20, position 1 to 2, which here position 2 is at 20 degrees. So if we look at the next, this is the pursuit. Maybe this will work. Try it. No. How can this be? No, okay, I just give up now. Okay. So here the eye is moving, follows this bird, say, from left to right, and that's just the same as I showed you in the slide, my previous slide. Here's the bird moving. There's a catch-up reaction time, saccade that catches up, and then the eye stays bang on target. In the vergence movement, the target's moving this way, the two eyes are moving in opposite directions, and these are called the vergence system. And this is the vestibular ocular reflex, where the head is moving in one direction. Both eyes together are moving exactly in the opposite direction through the semicircular canals. And now, having overviewed that, obviously the eyes are moving, and they're moving because there are muscles that are pulling on them. And the musculature responsible for moving the eyes is also reasonably well understood. And it's composed of 3 pairs of muscles that each lie in 3 orthogonal planes. 2 muscles per plane, 3 planes, 6 muscles. So this is a top view of the eye here, where this is the nose, temporal bone here. This bottom is a lateral view of the eye, which the temporal bone has been cut out. Going back to the top one here. So let's look at the different muscles. The horizontal movements of the eye are done by the lateral rectus muscle, labeled here, and the medial rectus muscle, labeled here. The lateral rectus muscle is seen here in the lateral view right here. So you can obviously deduce that the 2 lateral and medial rectus muscles are involved in horizontal displacements of the eye. Vertical movements are a little more complicated, and they truly are more difficult to understand. Vertical movements are activated, say you're looking vertically up, by 2 pairs of muscles here, shown as the superior rectus muscle here, and the superior oblique muscle here. Now this muscle is slightly inclined forward, so it tends to move the eye downward and rotates the eye about the visual axis. And this muscle is counteracted by the inferior oblique muscle, which is sitting down in here, shown here. So that the planes of the oblique muscles are in the same plane, I should say, and that plane is orthogonal to the plane of the horizontal muscles and also that of the superior and inferior recti muscles. Interesting, this is an interesting muscle right here, if I can find my little pointer. You see that this insertion of the muscle is so close to the nose that it doesn't allow for the belly of the muscle to lie along it, along this position. So nature has devised this clever little, you might call it a pulley or a sheath, in which the tendon wraps around the sheath here, right at this point, and the belly of the muscle is here. So that the action of the muscle is in this on this axis, but the belly of the muscle is here. Now let's consider where are the motor neurons for these muscles. Now, motor neurons are the neurons that actually activate those muscles. The lateral rectus muscle here and here down here, the motor neurons are sitting in the abducens nucleus, the sixth nerve nucleus right here. I don't know, I lose my pointer occasionally. The motor neurons in the fourth nerve nucleus activate only one muscle, which is the superior oblique muscle here. All the other motor neurons are sitting in subnuclei of the third nerve nucleus here. That means motor neurons for the superior and inferior recti, for the inferior oblique, and for the medial rectus muscle. And I'll explain how that works later. So the medial rectus muscle, I recall to you, is this muscle sitting here, which together with the lateral rectus drives horizontal saccades. Now the lateral rectus is driven by motor neurons, as I said, in the abducens nucleus right here, but its counterpart, its opposite muscle, the medial rectus here, its motor neurons are sitting in the third nerve here. So these two nuclei, the sixth and the third, have to communicate with each other. Feel free to interrupt and ask if something is unclear or you don't see where I'm going. So to summarize the organization of the extraocular muscles, 3 pairs of muscles. Each pair is in a plane. Each plane is orthogonal to the other. This organization, you might call this a footnote I'm going to make now. This organization probably developed from the original need to stabilize the eye in space during head movements, which we saw as the vestibular ocular reflex. Because the vestibular ocular reflex was the first of the eye movement systems to develop in nature. Millions and tens of millions of years ago. The reason, as I mentioned, is that the first job that nature tried to do is make sure that your eye compensated for head movements, even in animals that don't have a fovea, to prevent the image from sliding across the retina every time the animal made a movement. So nature, through its cleverness, developed these abilities to stabilize the eye relative as the head moves in space. And the saccadic system and other systems were tacked on, so to speak, by evolution later. And then another part of the organization, the neurons are sitting in the third, fourth, and sixth nerve nuclei, and those are easily accessible by microelectrodes, say in the alert monkey, if you're trying to understand the circuitry. So here we'll focus on saccadic eye movements, which are rapid displacements of the visual axis in space. I've said this. The only type of eye movement we can do voluntarily in a spatially stationary environment. Now, that some people say, oh, I can move my eyes slowly. But in fact, you can't voluntarily. If you think you're moving your eyes slowly, what you're really doing is making a series of very tiny movements. But if you don't believe it, you can ask your neighbor and try to move your eye very slowly in space, as slowly as you can, as I'm trying to sweep here across the room. But actually, you're making just a series of tiny saccades. There are subjects that have trained themselves to move their eyes slowly. We tend to call them ocular motor athletes, but there's no known competition for this. So now I've put here this system, the saccadic system, demonstrates that to understand cortical ocular motor control mechanisms, it is necessary to understand the mechanical properties of the orbit. This is actually quite important. As I said before, I'll go into this later, I think. Yes, exactly. So this is just to summarize. We're going to talk about saccades, rapid displacement of the eye, just to show you that this is omnipresent. This is one of our favorite cats, which we measured the eye movements in. You can see that cats even have nice little saccades, both horizontal and vertical. Now here's a schema of the cortical structures and their subcortical projections that are responsible for driving saccades. So here we can begin by pointing out that in the pons here sit the motor neurons that drive the lateral rectus, the medial rectus, and the other muscles, pontine reticular formation, you'd call it. This area, the pontine reticular formation, of course, sends the signals to the eye muscles. How does these neurons that are sitting here get activated? They receive a number of signals. I mentioned cerebellar inputs, which we know adjusts the neural signals in case there's, say, an injury to the muscle. But nature didn't only put the cerebellar input to compensate for injuries because they're quite rare. The cerebellum is thought to actively involve saccade control in ways that we still don't quite know. For example, it might be involved in stopping saccades. We know how saccades are start, but it's a little more complicated how they stop. Now let's get on to the cortical control. We can divide them roughly into the visual or sensory inputs from visual cortex and parietal lobe, which the structure that's been most studied is area LIP, which is lateral intraparietal sulcus, LIP. The frontal lobe structures are very important in driving saccades, namely the frontal eye field, the supplementary eye field, the prefrontal cortex. Now we know from experiments in monkeys that if you lesion the superior colliculus and the frontal eye field, you abolish eye movement control in monkeys. Monkeys cannot make saccadic eye movements. And the reason is because all the structures project to colliculus. As you can see, it's like a funnel here, where that's the final point in the trajectory of the parietal and frontal projections. Except for one thing, that the frontal eye field projection actually bifurcates and has an independent projection to the motor neurons. So if you just remove the superior colliculus here, then you get rid of a lot of visual-driven saccades, which monkeys have trouble making, but not saccades made for other reasons. For example, as you're thinking of looking somewhere, you're talking to a friend and you're making movements back and forth, so forth, which would primarily be driven by frontal structures. Okay, so there's different systems depending on whether they're sensory driven or whether they're voluntary. You might cognitively driven, you might say. So we're going to start at the bottom of the chain, right down in here in the pons. That's where we're going to spend a lot of time, or a lot of slide work, I could say, just trying to tell you what's going on in the pons. Because, as I said, once you've defined what the circuitry is in the pons, what the neurons are doing there, then you can now start to go uphill and see what's going on in the superior colliculus, which, as you know, is in the final common pathway, and then try to understand better how the superior colliculus is talking, so to speak, to all these premotor circuits or premotor neurons sitting down in here. And then once you know that, you can start now trying to understand how the cortex is is generating signals, the organization of the cortical descending pathways that are able to match the requirements of the midbrain with superior colliculus and pontine reticular formation, which is where the early elementary circuitry is elementary in the not in the simple sense, but in its hierarchical sense. So let me just continue now. So we're going to look at discharges in the pons to begin with. And we're going to first look at motor neurons and the force they must generate to move the eye muscles. Now this, as I alluded to at the very beginning, the major input or start of this work was made by a professor at Johns Hopkins University in 1964, a long time ago, called David Robinson. And one of his early papers, which had a huge impact, was called The Mechanics of Saccadic Eye Movements. David, I call him David because I was in his lab when I was a young guy, for a while at least. He reasoned that to understand motor neuron and brainstem and cortical saccade-related discharges, he reasoned that it is critical to understand the forces required to move the eye, the so-called plant mechanics in engineering terms. Now, Robinson was a control system engineer, and his work, early work in describing the forces that were required to move the eye by studying the mechanical properties of the orbit where the eyes are sitting, that was perhaps the first major contribution of what you could call computational neuroscience. It was a mathematical approach to defining a circuit, and indeed, this is Robinson's original circuit that he drew himself, and this is the motor neuron sitting here, MN over here. Based on his studies of the mechanical properties of the orbit, and known properties of the superior colliculus, Robinson was able to propose a circuit diagram with the existence of all these neurons that project to the motor neurons, and the existence of what is called a neural integrator, inhibitory neurons that prevent non-purposeful saccades, so forth. This entire circuit is based on one approach, which is to measure the mechanical forces that are in the orbit that are required to generate saccades. And like I say, it's a remarkable contribution, which is really one of the original major ones of computational neuroscience. So we're going to go through this, how we obtain this reasoning. So we'll go through this in the next few slides. So Robinson asked, well, how can I measure the forces that are required to move the eye? He did that on human subjects, normal subjects, at the beginning by putting a contact lens on them by suction, which held the eye very tightly to the cornea. And he applied different forces because these lenses had a little rod that was sticking out, and he could apply forces to the eye to you might say the studied eye, which was blanked out because the subject could only see from the good eye. So if the subject looked straight ahead, you could apply different forces and figure out what forces are needed to move the eye. But this is like very complicated, and these are like there's a couple of papers on this. But he followed that up with much more simple experiments, which I think are telling in their own right. And what he did, he had a buddy who was an ophthalmic surgeon in California. And this surgeon's specialty was to operate patients that had strabismus. Now, I don't know if you know what strabismus is. It's a condition in which the two eyes are not parallel. The axes of the two eyes are not parallel to each other. And you find that there are some kids, I don't know the percent of kids that are born that way, and now they're operated on very quickly. In the older days, some people they were just not operated at all, and they grew up and they had their two eyes were misaligned. So that had two problems. One, first, the major problem is, of course, you lose the vision in the eye that's deviated, and it's called amblyopia. And the brain simply just decides it's going to neglect that eye because it's not pointing in the right direction. Another reason was people were operated for strabismus for a simple cosmetic. People didn't want their two eyes. They wanted to have their two eyes to look straight ahead, basically. So these experiments, which I'm going to just show you here on this slide, was done in an adult patient lying on his back. There was on the just above him, they had set up a screen, which little target lights, which the subject could look at with his or her, I can't remember, a good eye. The bad eye was covered, but they had removed the muscle which was responsible for the eye being for the axis was deviated. And the surgeon's job, he was going to actually take that muscle and sew it, attach it to the globe, the ocular globe, in a different position, so that eye would be parallel to that eye. So the surgeon had, just after he had severed the insertion of the muscle on the globe, they put a little device. They actually took that muscle and put a little strain gauge on it, and measured the force in the muscle, the tension in the muscle, as the good eye was looking around. Okay, so then they could then plot, as shown here, they could plot the muscle tension here versus the eye position here. And this is done in the medial rectus muscle, because you could see that first it's because the force increases as the subject looks nasally, but you could see that roughly the tension in the muscle is proportional to eye position. It got much more accurate, but roughly this was a confirmation of his earlier work. So you can begin by writing that muscle force is proportional to eye position in steady state. Then they looked at the strain gauge result while the subject was making saccades with the good eye. And of course, since the bad eye is trying to follow the good eye, the two eyes are moving together, they could measure the tension in the good eye while in the bad eye, on the muscle of the bad eye, while the good eye is making a saccades to different targets in the screen above the patient. You could yes.
Is this person awake while they're performing eye surgery?
No, they're under sedation, but you don't really need to put them to sleep, because actually you're really only cutting. It's not that complicated surgery actually. What's complicated about that surgery is not to open the eye, not the eye, but the tissues around the eye, and expose the muscle. What's a little more complicated is where you're going to sew that muscle. You cut it where it is, but you want to sew it somewhere else, and you better be right in sewing it, because the reason you're removing it in the beginning is because the person's eye is crooked, right? It's not crooked. The visual axes are not parallel. Okay, is that clear? So you can see right away that from just by scanning here, just by intuitively, if you look at the profile here, the force profile here, it resembles the velocity of the eye. So you could roughly write, we're just showing you ballpark approaches here. You can roughly write that the force in the muscle right here is proportional to eye position and eye velocity. That equation was the basis for huge advances in that circuit I just showed you earlier. Now, I didn't read the top part, but we have considered motor neurons and the forces they must generate in the eye muscles to produce saccade. We now show that eye muscle force is equal to a constant times eye position plus another constant times I velocity, and I've lost completely power in my little pointer here. So next question, given that you now know the forces, Robinson said, well, I'm going to record in the alert monkey. So that he started recording the alert monkey. I would say that it was probably in the early 60s. And one of the first people to record from the alert behaving monkey. And what he did was to put his microelectrodes in one of the three motor nuclei, which had the motor neurons for the muscle. And they went into this third nerve nucleus because, as I pointed out, there are many motor neurons for different muscles here. And it's a big nucleus. They could have tried the abducens, which is possible too, and in many ways simpler, but that's a smaller nucleus, contains the motor neurons for the lateral rectus muscle, but it's the lateral rectus muscle, the action of the muscle is well understood. Would have been a little more complicated to go into the fourth nerve nucleus here, because that activates the the superior oblique muscle, whose role is quite complex in terms of directions and calculations of it, pulling directions, and so at different eye positions. So that the easiest is to go into the third nerve nucleus, which is what they did. And here's some examples of their findings. This comes from a paper written in 1971. And so when the monkey is looking straight ahead, you could see that the motor neuron discharge from a neuron that is in the third nerve nucleus that relates to vertical up or down motion or positions of the eye. But this neuron, as you could see has very steady firing. If you now look at this neuron's discharge during as the monkey looks at different positions in the room, and you plot the discharge here of the neuron, discharge rate in impulses per second, versus eye position. This neuron at the points shown here, you see that there's a linear relationship between the firing frequency of this neuron and the eye's position. And this relationship is different for different motor neurons. And of course, there are many motor neurons, so the full signal is the sum of all these, and that's still a complicated problem to resolve because they have different thresholds when they come on, as you could see here. You see, like the neuron B here comes on when the eye is deviated to the left by about, say, 10 degrees, whereas these neurons are silent. Neuron D only comes on when the eye is looking about 25 or so, 23 degrees to the right. So they have different thresholds. But at least for any one neuron, you have a linear relationship between its firing and the position of the eye. Similarly for velocity, if you plot the eye velocity and the discharge rate of a neuron, you find that that same neuron discharges linearly with eye velocity. It's kind of neat if you're recording in this motor nucleus, if you take the neuron's discharge and place it through an audio amplifier, you can actually, it's like you can hear you can call it the singing nucleus, because as the monkey looks around, the frequency changes. So you can hear, you can almost like you can go, and so just by hearing, you know where the monkey's looking. That's an anecdote. So now we have two equations. One is the first one I showed you, that the muscle force is proportional to eye position and eye velocity, and that the firing frequency of a neuron, any one motor neuron, is proportional to eye position and eye velocity. So therefore, firing frequency is proportional to muscle force in the ocular muscles. Not say this is is being extended to your arm muscles or leg muscles, but in the motor neuron, in the ocular motor system, that's an approximation. It's more complicated than that, but you can understand a lot of what's going on by just knowing these two equations. So let's move on now. So as I said, just to remind you now that the neurons have this burst of activity, or I'm sorry, the tension has like a burst-like property during saccades, proportional to velocity. And indeed, now when Robinson went into the motor nuclei and obtained the results I just showed you, if you look now at those motor neuron discharges during saccades, here's what you see. So this is an eye movement. As I said, he's in the third nerve nucleus, and this neuron responds to downward motion. So the eye starts at 20 degrees up here in this example and moves in a saccade to 20 degrees down. And this is the discharge rate of a typical motor neuron that is sitting in the third nerve nucleus and controls vertical movements. So you could see that which you can now predict, I'm sure you know, that you have a burst initially that's proportional to eye velocity, and so the saccadic eye velocity is very high. So you have this nice burst of activity, and then it settles to a steady state discharge, which is proportional to eye position as the other part of the equation. Okay, is that clear? Now, if you look at the same neuron, but the eye is going in the other direction, so it's going from here now to here, what you see is the eye starts here with the discharge approximately equal to that, but the eye is not exactly looking, I'm sorry, approximately equal to that. This is not exactly at the same position here, but you could see that the motor neuron is completely silent during and resumes at a slightly offset position at a slightly different frequency because it's at a different final position. Now, one of the things that's noteworthy in all this is that there is no breaking pulse. So you can think of, if there was breaking, at the end of this saccade, you'd expect that this neuron would have a little burst to break the saccade. It's going down, but there's no break. And here too, you see, there's no perception of a breaking pulse. And that is compatible with the other measurements that the forces are proportional to eye velocity, because you can think of the eye as being a balloon that's rotating in a barrel of crude oil, say, or in Quebec here, a barrel of maple syrup. You could see that the eye, you can think of the eye as just a weightless balloon that you're rotating in a very viscous environment via two strings on either side. And so that immediately is the picture of what the brain is trying to do. Is the weight of the eye, so-called its inertia, is negligible. Robinson actually looked at this by putting different weights on the eye. In the experiment I told you about, I didn't want to get into detail, where he has a rod sticking out attached with a contact lens to the eye, and you can put weights on the rod. That's it's not a very pleasant experiment, also quite complicated. But the fact is that he did put weights on, and it doesn't change the trajectory of the eye. So that therefore, if you're rotating a balloon in a barrel of maple syrup, as soon as you start pulling, the balloon stops. And this is very different than if you're thinking of limb motion, for example. And if you want to move say you want to flex your arm like this, to flex your arm like that, you have to activate your biceps. You need a big burst in the bicep, boom. But because of the inertia, you need a braking pulse in your triceps. So I don't know if you've got that yet in your course, but you burst, you brake pulse here, and then because of the weight of the arm, it tends to oscillate. So you need another little burst right in the biceps again to prevent the oscillation. The eye, no worries. Never worry about that stuff. You pull it, it moves. You don't have to break it. As soon as you stop pulling, the eye stops. That's why it's kind of a convenient system to study. Okay, so moving on, that's a summary, a functional summary of what we have for saccades. You could see that there's the eye is going from 1 to 2, saccade right here. Oops. The eye starts at one frequency here, steady state. The eye is just looking steady. And then at this position, the frequency is higher just to hold it in position. And to move the eye rapidly, you have a pulse. This is an idealized pulse. You saw it before that it's not quite like that because it's more proportional to velocity. But this is a satisfactory approach or to understanding the system. You need a pulse step to drive the saccade. If you don't have a pulse, this is what happens. If you just increase suddenly the firing frequency, the eye moves from one position to the next, but very, very slowly. And of course, you're not going to survive much in the world if you take such a long time to move your eyes to get it from one position to the next. So, so now we move to the next step, which is, but by the way, maybe I should ask formally, are there any questions? All clear, I guess, or it's completely impossible to understand? Two alternatives here, okay? So this is the schema. I put that in because we can now divide and recall that Robinson had made this diagram, and he suggested in that diagram that this pulse here, so you have the saccade, this is the pulse step here, but this pulse, he, based on other studies, which I can't get into, he suggested that this was produced by an independent set of neurons that projected to the motor neuron pool. And he had reasons to believe this based on the mechanical measurements, not by his recordings. So based on the mechanical, he realized he proposed that this burst here is proposed, or that pulse is produced by a different set of neurons, which we call burst neurons. And so this we've shown in a private side. Now we're going to show neurons responsible for motor neuron discharges. They're in the paramedian pontine reticular formation, burst neurons, and omnipause neurons. And then we'll move on to superior colliculus. This is how we're going to proceed now. So here we go. Now we're back to the brainstem and the recordings in the monkey. And this time we're going to be in and around the abducens motor nucleus. That's the lateral rectus muscle. Moves your eyes in the temporal direction. The neurons, the burst neurons that Robinson proposed must exist to activate the motor neurons, these burst neurons are sitting in the paramedian pontine reticular formation right here. This area here is responsible for driving horizontal saccades. Vertical saccades are the subject of interest of this region in the reticular formation. The third, fourth nerve nucleus. This is called the interstitial nucleus of Cajal, mesencephalic reticular formation, interstitial nucleus of the medial longitudinal fasciculus, nucleus of Darkowitz, can't remember quite how to pronounce it. But all these neurons now are involved in the vertical movements. It's a little more complicated than horizontal. So we're going to be sitting down in here in the next few slides, okay? So we're going to talk about burst neurons and pause neurons, and I'll tell you what they do. Pause neurons. So here's two examples of burst neurons sitting here on top. This is a burst. This is the saccade, horizontal, because we're dealing in the horizontal system. And this burst is called a long lead burst because you might call it a preliminary discharge that precedes the main burst. These are burst neurons, the clear burst neurons where there's only a burst, okay, that relate to horizontal. There's a slight here vertical component, but the burst is related to the horizontal. And then and this is a pause neuron. Pause neuron because it stops firing during saccades, and it has a steady state discharge otherwise. So here we start and I'll show you the typical discharge of a burst neuron. So this work was done by Dr. Kathleen Cullen, which was in the physiology department, is now a professor at Johns Hopkins. And here's a couple here's one burst neuron, and for two saccades. There's a small saccade here, larger saccade right here. This is the I velocity for each saccade. Now you might obviously realize why we're showing I velocity, because you know that the pulse created in the motor neurons, created by the burst in the burst neurons, that the pulse is proportional to I velocity. And true enough, if you look at the firing frequency of the burst neurons for the small and large saccades, they can be nicely fit by an equation, which is written here, that the firing rate is equal to a constant plus another constant times I velocity. And if you plot number of spikes versus saccade amplitude, then you get a relationship which is linear, which is obviously would be the case. The number of spikes is the integral of velocity, and the integral of velocity is position. So you could, knowing that this equation here fits the burst neuron discharge, you will predict that the number of spikes in the burst are proportional to saccade amplitude. The omnipause neurons now, these are neurons that are sitting right on the midline in the reticular formation. The brainstem midline. This is the motor neuron, say, for the lateral rectus muscle. We're going to get into a full schema later, but so these are just elementary schemas. We'll get into the full thing with my ancient instrument. So the motor neuron, sorry, I'm hesitant because sometimes the arrow just disappears. This is the motor neuron. As you know, it's a pulse step discharge. The pulse is driven by the burst in burst neurons. This is an excitatory burst which activates the pulse step discharge in the motor neurons. There's the excited burst neurons are also known to excite other burst neurons, which themselves burst and inhibit the contralateral muscle. So on one side, you have the lateral rectus on one side is being pulled, but in the other eye, the lateral rectus muscle must be inhibited because it's the medial rectus muscle of that eye that's pulling. So again, if I'm looking straight ahead and I'm looked that way, then my lateral rectus muscle here and my medial rectus muscle on this eye both have a burst step. Boom. But that means that this muscle now, the lateral rectus muscle here now is now the antagonist muscle, and it must be silenced. And it's silenced because of the inhibitory burst neuron here, which is inhibitory by definition, and it just stops the discharge in that motor neuron. Now, these pause neurons are known to be inhibitory. That's been shown anatomically. And they sit and they inhibit these burst neurons. And the pause neurons, as their name implies, they pause, their discharge pauses during all saccades independent of the saccade direction. They always pause. So here, these are 4 saccades starting from the center. So you're up, down, right, left. And these 4 are for all these 4 directions. You can see that the pause neuron always pauses during each of these saccades. So now we're going to take our ladder and we're going to move upstream or up the ladder as we head to cortex. So now we talked about motor neuron discharges here, the burst neurons that are sitting here that provide the pulse that's in the pulse step discharge of motor neurons. We've talked about omnipause neurons, which also sit on the midline here in this region, which inhibit the motor neurons, the burst neurons. Now, the superior colliculus, as you saw from my earlier slide showing the cortical and subcortical nuclei involved in saccade control, that superior colliculus projects. By projections, I mean the neurons sitting here, they send axons into these different oculomotor areas. And the colliculus is known as the important relay station between descending cortical commands and the colliculus sitting between the descending cortical commands and the basic oculomotor circuitry, burst neurons, pause neurons, motor neurons, so forth. So the superior colliculus presents a very special challenge in understanding how a cortical descending input actually drives the motor neurons. So let's go on to this. So now we're going to talk about superior colliculus. So now if this is the superior colliculus shown here as C, okay, we're now going to record here. Now, if you take your each and every one of you must have his or her own brain image. And if you take a cut through your thing, you do it here. This is the colliculus sitting down here. And this is what you see. There are two, one on either side of the midline. The midline's here. Whoopee, back here. And the colliculus is a layered structure. Layer means that there's alternating layers of cells and white matter, fibers. Cells, white matter, cells, white matter, cells, and so forth. The superficial layers of the superior colliculus belong to the visual system. They're the part of the primitive visual system. They have axons which project upward through the pulvinar into the extrastriate visual areas. The intermediate and deeper layers of the colliculus, the neurons there are involved in driving saccadic eye movements, and even in the more deeper layers, driving associated head movements. And even deeper, there's evidence that they also control arm movements to targets that you're going to look at. But that's like really complicated stuff that we don't have time to look at. So let's look at the colliculus now in more detail. This schema, this is a schema. This is just what I showed you here just a second ago, the previous slide. If you cut away the superficial layers on this side, you reveal the motor layers, as I said. Whoops. And on this side here, the superficial layers are shown intact. And the schema here shows that both the superficial layers and the deeper layers contain maps. Here, this is a motor map. If I can this is a motor map, and this is a visual map. Now, the visual map here is similar to the visual map that you see in primary visual cortex. So it's what is called a retinotopic coding of visual space right here. And sitting right below this retinotopic coding is a motor map, also retinotopically encoded, which sits just below it. Pardon me. We can get into more detail on this in a second. So this map here, motor map, contains motor neurons and fixation neurons. The neurons on the motor map, the motor neurons, will encode saccades of different vectors, different directions and amplitude. For example, this is the horizontal meridian. All saccades controlled here are horizontal saccades. If you have a discharge here on the map, this would control a horizontal 10-degree saccade. If you have sorry, I got these delays here. If you have a motor neuron sitting here, it would control approximately a 60-degree, this would control about a 60-degree horizontal saccades. The neurons sitting here, which are called fixation neurons, they have different properties. They are thought, some argument about this, but they are thought to be involved in steady state fixation because they put their anatomical projections are to the pons neurons. So if these are very active, the pons neurons are also active, and the motor neurons are inhibited, which you'd expect in a condition where you're fixating, you're looking straight ahead. Okay, so so one of the interesting things now about this organization, I point out right away, is that the vector of a saccade here on this map is encoded in spatial coordinates. The location of the burst on the map, whether it be for horizontal saccades or vertical saccades on these other parts of the map, the location of the active site on the motor on this motor map, its location codes the vector of the saccade. It's a spatial coding of the saccade vector. So if you have a burst here, 10-degree horizontal saccades. But recall from the previous stuff I was just talking about, if you have a burst in the burst neurons, the pulse in the motor neuron discharge, they're all coded in time coordinates, firing frequency, and duration. So we have the enigma here in the brain of how a spatial code gets transferred via these projections here into the burst neuron area, motor neuron and burst neuron area, that has a temporal code. Robinson, way back in the 60s, called this the spatial temporal transformation problem. You have to go from space to time. That's a tough problem, which there's still today not quite, there's a lot of argument about how this works. Don't forget, you can interrupt at any time. Huh? Okay, so these are, I just showed this schematically, these are real tectoreticular neurons injected by intra-axonal injections way back in the reticular formation. You could see that they're heavily, the dendritic trees are very large, and that the neurons here will cross the midline to the other side. So a burst on this colliculus will activate neurons here. Okay, so the right colliculus generates leftward saccades. Okay, I mean, yes, right, not saccades towards that's right in the opposite direction. So that's these neurons. Now, if you want to follow these neurons downstream, these are labeled neurons. These are not in the monkey; they were labeled in the cat. But you could see. So this neuron here, or axon, originates in the colliculus, and you see it's moving caudally towards the spinal cord, and it's dropping off axons, partly in the abducens nucleus, in the sixth nerve nucleus, but also in reticular areas, which contain the burst neurons over here, and other neurons here in this prepositus hypoglossy nucleus of the brainstem, which contains the neurons which integrate the velocity signal that's in the burst neurons. And I'll show you that later when we do our schema. This is a proposal by Robinson where he proposed that you need a neural integrator, and so these neurons project there too. And then they have all these other complex projections. So this is just to illustrate that when you're recording neurons, you realize how complicated these projections are. It really raises your either worry level or you want to head to the bar, because it shows you how complicated it is to understand these circuits, even in a simple system like this. Okay, so now going back to the colliculus. The colliculus contains fixation and saccade-related neurons. Fixation neurons, as we pointed out, have steady state discharge, and they pause during the saccade. This is the saccade here, called gaze here, but it could be just a saccade. And then the activity resumes after. This looks very much like pause neurons, doesn't it? But they're sitting here on the rostrocollicular map, and we know I mentioned that they project anatomically to the pons neurons sitting in the reticular formation. So these neurons project to pons neurons, and they both have this type of discharge. If you look at it in more detail, there are things that are not quite the same in the discharges, but roughly speaking, they have rather similar discharges. Now, the saccade burst neurons, they burst during a saccade. Okay? Now, in the brainstem, we know that a burst before a saccade encoded saccade velocity, but this is not true in the colliculus because it's a spatial code, not a time code. So let's consider now the motor map. And so schematically now, it helps to be just schematic. So these are the motor layers, as we showed you before. Say you have a group of neurons here, which we can call a mountain of firing frequency, sitting at a particular location on the map, say about 15 degrees along here, perhaps off the line, midline, about 15 degrees down. The neuron sitting here will have this kind of firing frequency. You can see they have a burst. This is the frequency versus time. This is quite a nice discrete burst, except for a little preamble here. And then this burst will occur for saccades in a distinct region of the visual field. So this is a horizontal axis, vertical axis to the right, to the left. So this corresponds, this activity pattern here corresponds to, where are we here? To this here, this hill of activity, of firing frequency. And here, if you look at the firing of the neuron shown by the closed circles and the crosses, this neuron will fire for only movements that go from here to here, but not for movements anywhere else in the visual world. So if you start here and go to here, neuron silent, here, go to here, neuron silent, here, go to here, neuron silent, etc., go to here, silent, but here, you got a burst. Okay? It responds only for a particular group of vectors, gaze vectors, to a location in space, in retinotopic space, relative to your fovea. So here's a schema now of what's going on in the collicular map. You start by activity in the fixation area shown here, and this is the progression from making a saccade to the end of the saccade, schematically. So at the beginning of the saccade, you question, maybe I'm going to make one or not. There's just low-level activity where you may want to look. And then as you say, yeah, I'm going to look, the activity builds up here, from here to here. Meanwhile, the activity in the fixation area goes down, and this generates a motor command to drive the saccade. And at the end of the saccade, you have activity now back again in the fixation zone. So pause of activity here, as you saw, during the saccade. Now the question then is that activity at the beginning of the saccade, activity starts here on the map. At the end of the saccade, the fixation zone is active. So now what happens? So let's do this schematically here. At the beginning of the saccade, so you're driving, you want to make a 50-degree horizontal saccade. You have a nice burst here on this map. And then at the end of your saccade, you have the fixation area that's active. So question now, how does activity go from here to there? One idea that a student of mine proposed many years ago is that the activity actually slides across the map. It's called the moving hill hypothesis. So the idea is that during a saccade, activity starts here and just slides along the map, probably due to feedback here on the interneurons, which are pushing this activity towards here, the fixation zone. And once the activity reaches the fixation zone, the saccade stops. So you could see this here like that. Okay. So, oops. It doesn't jump here. I should have made it continuous, but it just illustrates that the activity just moves from one site, the motor area, to the fixation area. So now let's overview the saccade circuitry. This is the original drawing I showed you before by David Robinson that he made way back in the 60s. He had this motor neuron discharge, which was the pulse step discharge, which I described. He, through his measurements of forces, he argued that the pulse here needed to be generated by an independent set of neurons that were called burst neurons. These burst neurons project to the motor neurons and excite the motor neurons. They are thought responsible, are now known, but in those days, supposed to be responsible for driving the pulse step in the motor neuron discharge. Now, these burst neurons encode velocity. But we know from what I showed you that the motor neurons at the end of the saccade code eye position. So you have some neurons that code eye velocity and other neurons that code position. How do you go from velocity to position? You integrate. So Robinson proposed that the burst neuron output, its output, projected to a set of neurons called the neural integrator. The neural integrator had as an objective to integrate the burst neuron discharge, create an eye position signal, because these encode velocity, integrate velocity, get position, and that this neural integrator would project into the motor neuron pool, providing the position signal. That has been shown to be true. People have found where the neural integrator is in the reticular formation. It sits in a nucleus called the prepositus hypoglossy nucleus, not far from the vestibular nuclei. Burst neurons, I talked to you, I showed you, they lie in the paramedian pontine reticular formation for horizontal movements. But Robinson went a little further than this. He realized that the burst duration here changed as a function of the saccade amplitude. So you needed to have a control on the duration of the burst. That was a problem. How did you stop saccades? Because the saccades would stop if you ended the burst discharge. He also realized that you had this spatial temporal transformation problem, which goes from the collicular map, which is spatially encoded, to the reticular formation, which is temporally coded. So he argued that this could be done by a feedback loop, which compares the desired eye position, here, theta desired, the desired shift in the eye, the desired rotation of the eye, which was provided by the superior colliculus. But then to control the duration of the burst, you also had to make sure that the burst would stop at a certain point. And the way he managed that, he proposed a feedback loop onto here, an inhibitor interneuron. So this feedback loop would actually inhibit the pos neuron as soon as the desired eye position had the desired eye position had met the. Sorry, let's start again. As soon as the final eye position had met the desired eye position. And how do you get the final eye position? By the output of the integrator. So basically what he's suggesting is that you have an actual position signal on the motor neuron, but also this estimate of eye position is fed back here in the feedback loop to an inhibitory neuron, which will inhibit this burst neuron until theta estimate is equal to theta desired. And once these two are equal, this burst is no longer excited and then stops discharging by here, and the inhibitory signal on the pos neuron stops, and lo and behold, the saccade is stopped. These are trigger signals, which are another part of the requirement. But just to think now, just on the basis of mechanical measurements and the knowledge that you had the colliculus coded in spatial coordinates and the brainstem was coded in temporal coordinates, he could come up with a proposed schema like this, which I and many others regard as being a major step in what we call computational Dave. Another question, sorry. Neural integrator. Yes, we know that because the burst neuron, I got your question right, tell me. If the burst neuron codes velocity, right, we know that the motor neuron burst, the pulse in the motor neuron discharge also encodes I velocity. That showed you in the previous slides. So that, and we know now from anatomy and so forth that these burst neurons project here. But they code I velocity. So the way you get I position is to integrate I velocity. So this was the job of the proposed neural integrator, which was initially theoretically proposed, which was then confirmed by experimental observations. Is that the answer? Huh? Yes, that's proposed. It's sending branches here and also here. And you might say that this is not too surprising when you saw the tectoreticular neurons and their vast projection tree. And these burst neurons have similar varied projections. Any other? Oh.
What's the experimental evidence to support?
Well, it's quite simple actually. First of all, people have recorded in this nucleus, which is called the nucleus prepositus hypoglossy, sitting in the brainstem. Okay, and they've recorded extensively through from the input stage to the output stage of the nucleus, and found that there's a gradation. So neurons at the beginning discharge to velocity, and at the end they're discharged proportional to I position. And then there's been mathematical models of how this works in that nucleus. And then at the same time, if you take that nucleus out by lesioning it with reversible lesions, for example, then the monkey's eye position cannot hold. The monkey's eye cannot hold in a given position. Okay. Right? So, is that no further? So now this is a summary, which it's a summary of some of what I've told you. And after we go through this, then I'm going to go to my ancient machine here and try to reconstruct with you the actual circuit. Okay? And I hope that you'll find this a useful exercise. That's the reason I proposed to Dr. Ragsdale to resurrect ancient machinery, because I found that this little board here is so small that I wanted to have something that's very big and you could just all see it no matter where you are. Okay? So now let's, excuse me, let's just overview on this rougher, but many of the important points are there. Okay? So this is titled The Superior Colliculus and the Pontine Reticular, Paramedian Pontine Reticular Formation Saccade Generating Circuit. Okay. So now this is a cut through the skull, so to speak, mainly the reticular formation. And this is the right eye looking to the right. Left eye is looking to the right also, conjugate saccades to the right. Now, if you look in the lateral rectus muscle here, you must have activation here. And here we know that from the data I showed you that this neuron is inactive during rightward movements. It's the antagonist neuron and it's not active. And similarly here, the medial rectus here has this discharge and it's suppressed here. The discharge in the motor neurons in the lateral rectus is a pulse step discharge, as I showed you. Excuse me. In this neuron here, it's the opposite because it's the antagonist neuron. So this neuron, it just simply stops firing. And here in the medial rectus muscle, you also have a pulse step. And in the lateral rectus muscle, you have the suppression of activity. So you can see that both the medial rectus muscle here and the lateral rectus here both have this pulse step discharge. Okay? The question now is, what you need to build up is this is the abducens nucleus right here, motor nucleus. So this is where the motor neurons are that carry the pulse step are sitting in the abducens nucleus here. The question is, sorry, my voice is gradually slipping away. You need a pulse step in the medial rectus, and we know that anatomically that this is produced by interneurons that are sitting in the sixth nerve nucleus here, that these interneurons are projecting here across the midline, projecting into the third nerve nucleus, and which produce the pulse, which responds for the discharge here, and the pulse responsible for discharge here. Now, the integrator is sitting in the pontine, paramedian pontine reticular formation, as I pointed out, which the integrator in particular is in the prepositus hypoglossy nucleus. And so the integrator on one side has activity that ramps up and produces the tonic activity here. On the other side, it ramps down and it projects to the, it produces the diminished discharge in this neuron right here. Now, if we go to the collicular map here, as you saw, during fixation, these are active. The fixation neurons are active, and they pause during the. I'm sorry, I got this wrong. These are the fixation neurons, and these neurons pause during saccades right here, and the motor neurons of the colliculus that code the saccade have a burst. Okay. And these project into the pontine reticular formation, both of them, and they either say stop or move. Stop if it's the fixation neurons, move if it's these neurons. And that's the story in this particular view, and we can now look into more details about how this works down in here, in my little, if I can manage with my little ancient equipment. So, excuse me. If you want, you can presumably take a short break while I get this going, and we can convene again in. Yes, question. I'm not sure I understood. Now repeat. Why is the suppressed activity in the neuron that's connected where? To the right. To the right. Right over. You mean over here, right? Yeah, okay. So this is because this gets a relay signal from here. Now there's a difference. This neuron, this discharge here is due to activity here. There's a simple, I don't know how many just pure relay neurons there are in the brain, but this seems to be one of them. You have here the motor neurons that are driving this muscle, but there is a relay here from this abducens nucleus up to the medial rectus muscle, which is producing the same signal here as you saw here. Is that clear enough? No. Okay, so you want this side now. So the reason there is because during saccades, remember that I showed you this, if you make a saccade in this direction, the burst neurons on this side will burst, but there's a set of burst neurons which are called inhibitory burst neurons. They project to the other side and inhibit the motor neurons on the other side. Okay, so you would have an inhibition of the motor neurons that control the lateral rectus muscle, as shown here, right? And they're produced by inhibitory burst neurons that are sitting here. Okay, and then the interneuron here would also be inhibited, but there's no direct inhibition from in here. It comes from a lack of excitation coming from here. Clear? Yeah.
So, like the small squares that show the neuron activities throughout all the different pathways here.
Yeah.
So it's clear to see, like, when there's a spike, it's more active, and when there's a trough, it's less active. But like what is what is that graph actually showing?
Okay, let's be a little more specific. I'm not sure if you're following you. So let's take this here.
Yeah.
So what this is showing, sorry, I thought I didn't go over it, but I just assumed you got it from my previous slide. This is the firing frequency profile,
Okay.
right? If you had an electrode here in this nucleus right here, if you have an electrode in this nucleus, the axon projects right into the muscle here. That motor neuron has that discharge. You're asking how it's measured?
Yeah, like
It's measured by putting a microelectrode in the abducens nucleus. When the eye is looking in the pulling direction of that muscle, which you get this, which is a pulse step discharge, or if you're looking in the opposite direction here of this muscle, which would occur if you're recording on this side, then it would be it's inhibited because this muscle now is not the pulling muscle, it's the antagonist muscle. Not
Okay.
clear?
Yeah.
Okay?
Yeah. Yeah. Well, yeah, I think it's clear.
Any other points, or maybe because sometimes, I just assume that you got it and that wasn't clear. Any other? Everything cool for the moment? Okay, remember, like these are obtained from recordings in alert monkeys. Monkeys looking around, you put electrodes in these different areas, and you record, and you know where these areas are from the various atlases that encode where different nuclei are sitting in the brain. So we can record them, so you measure the eye position, which enables you to measure eye velocity. You record the motor neurons, you know where they're sitting, so you can deduce all this from this type of research. But it has to be done in an alert animal. So when you come back, I hope to have this set up, and then we can, like I say, go over the circuitry to begin with, and then we'll move on and consider cortical control of saccades as a second part, but which is not too long. Okay? Excuse me. Do this, and then I just play projecting loop.
So I think we should get started again, but there was something important I forgot to tell you guys at the beginning of class, which is that there's no justice in the universe because the evil Tom Brady and his evil overlord Bill Belichick won again. So there's no justice in the universe. I'm sorry. I'm sorry you had to learn this at such a tender age, but anyway. But I think that I forgot to tell you that. I think that's really important. And I'll carry on with the rest of your.
Why is Tom Brady evil? Yeah.
Yeah, that was a long story.
I'm a Tom Brady fan.
Oh, you are? Well, we'll have to fight it out.
You have to explain.
We'll have to fight this out. It has nothing to do with Donald Trump, so it has to do with Donald Trump.
Okay. Okay. Hope this is going to work. I'm nervous because this is like a new approach with old, what do you call it? The old and the new. An old approach, I drew it already. I could just actually show you this. This is what I'm going to draw. Okay, bye. Because that's what I'm going to end up showing. And happy with this, I'm going to go now. But otherwise, I thought that this is a little complicated. If I showed you this slide, you'd all fall asleep, or else you'd think of something else, or whatever, or else I'd have so many questions that I wouldn't know what to do. So I'm going to now start drawing this on a clean paper like this, and we'll see where we go. How's that? Maybe if I push this a little more this way. That's how it was done in the old days. So here, we draw the visual field. Let's just draw the half visual field. With that, this is a horizontal meridian, vertical meridian. So this is visual field, and say the target appears here, right? So that's a good start, right? So you're looking straight ahead, and then the target flashes here. So now we have to work out how this is going to look like in terms of the discharges. And the obvious place to begin with is to draw the eyes, because that's what we're considering. Right? This is the midline, and there's going to be an eye here. And there's another eye here. Okay? And so the eyes are both going to, now we're assuming, of course, that the eyes are very, very far away in the visual field. Otherwise, if they were just as close as that, you'd be involved in convergence movements and stuff, right? So we're assuming that these eye movements are now very far apart. I mean that the eyes are very far away. So that this eye will move like this. And since the two eyes are moving together, this eye will also move here. And then both eyes have muscles. Okay. Now, this muscle here is what muscle? Do you recall? Lateral rectus. Sorry, I can't see much. And so it's the lateral rectus on this one, and this one is no lateral rectus. Aha. This is the medial rectus, and this is the medial rectus. So we know from what I've just told you. Make sure I get this all sorted out here. So we have here. We'll draw the motor neurons. I send a projection this side, we also have motor neurons that are sending, right? And then we have medial rectus mostly, and here. Okay? And this medial rectus that way. That looks like this. What does this discharge look like? What do you see as a discharge here? And, sorry, I drew my line a little too close to it. What do we have here? I'm going to draw the firing frequency profile. What is it? Burst? But there's more to it than that. There's a burst here. That. What precedes the burst and follows it? Sorry? What is, if you're recording in a motor neuron for this saccade, what do you see at this point? Burst like this also? Huh?
Slowly increase.
Sorry?
Slowly.
Yeah. So you have here, you have tonic activity before, and the tonic activity in the pulse step discharge pattern, right? The activity here needs to be higher than here because the eye has changed position from here to here. So the pulling on this muscle here must increase between this position and that position. This is what you see here in the motor neurons on this side. Okay, is that, just check here, it's not bad, with blinding this. This is what you must see in this muscle right here, in the medial rectus. You also need this same discharge. And where does that come from? It comes from other neurons that are sitting here in the same nucleus. So you have, so sitting here, the which is the sixth nerve nucleus, has two types of neurons. One type projects directly to the lateral rectus here. The other type, interneuron, projects to the medial rectus motor neurons. And both of these neurons have this discharge pattern. We're on this side now. What do we see? So we see that we have the saccade. The saccade is going same saccade now. Okay. Like that. And what do we see in the neuron discharge? Inhibition. So we go here, and then the activity simply drops to 0. Okay? Because the activity here at the end of the saccade, is it going to be more or less than here? What? No, can't be the same because the eyes move. Less. Right? It has to be the mirror image of this. The eyes, for this muscle, the activity increases, needs bigger discharge here because the eyes change position. It goes from here to here as it goes from here to here in the visual field. So this eye now is the discharge in this muscle has to be the same as this muscle, but this muscle, the discharge is decreasing because moving in the off direction. So at this point, right at the end of the saccade, the activity is less. All right? That's what you see if you're recording with microelectrodes in the alert monkey. Excuse me. So now I'm going to be sure that I get this more correct here. Change color. To the brainstem circuitry. explaining that we have to recall to you that we have, it's always crossed, right? So there are two neurons here. This neuron here has, this muscle here has the same discharge pattern. Muscle has the same discharge pattern as here. What we see right on the midline, right below it here, around that, it's true. Anatomically, it's true. In the brainstem, right close to these neurons are other neurons, the pause neurons. And making them full, they're inhibitory neurons. Pause neurons are inhibitory neurons. We have excitatory burst neurons that are sitting here. We call them OPNs, and when we show right. And what's the discharge of OPNs for a saccade? Have a tonic discharge before the saccade. During the saccade, they stop discharging and they pattern of the first neuron. First neuron projects right into the motor neuron, right? Like that. What's the discharge characteristic? I know myself. That's the verse neuron. EN, EBN. Pause neuron. I showed verse neurons. What does it do on the verse neurons? So I have to look because there's so much in there that if I don't I have to lay it out properly. Otherwise, now these burst neurons project other neurons sitting here, which are inhibitory burst neurons project, right? Excitatory projects, they drive the pulse here, and these guys inhibit this neuron here to, so these guys project this way, like that. Assuring that there's a pause here because this muscle is the antagonist muscle and it has to be completely relaxed since the eye is moving to the right. Okay. Now the next step in this, take the output of the burst neuron, you have to put it into another circuit here. Box here, I'll just call it, there's a box here. This box connects back into here to the absolute nucleus. What's this box? Integrator, someone said? Yeah. I think so? Yes. We call this a neural integrator. And that's sitting in the nucleus prepositus hypoglossy nucleus, and it's excitatory. So the output of this is low-level activity here. During the saccade, it ramps up, and then there's tonic activity here, like that. So you can now see, okay, how this burst or pulse step discharge is created, right? It's created by two things. One is the excitatory burst neuron input into the motor neuron pool here, which is a pulse. The pulse is sent to the neural integrator. The neural integrator will integrate this pulse. Now the frequency of the burst neuron pulse, what is it related to? It's proportional to what? Eye velocity. So if you integrate eye velocity, you get eye position. So this little circuit produces this motor neuron discharge, which is a signal proportional to eye position, and the signal here proportional to eye velocity. The pulse step, the pulse is necessary to make the eye move very fast. The pulse is proportional to eye velocity. Okay? Now on this side, you need the mirror image of this. Because remember, you have burst neurons here, but you also have burst neurons sitting on this side too. They're probably done, I run out of ink here. And I just got to get this right because I want to begins. Yeah. So the IBN, which is sitting here, which assures, is that clear? The IBN, yeah, right, it projects there. The IBN, just like this EBN, the IBN also necessarily projects into the neural integrator. But this time the activity actually this one inhibits here, like that also, negative sign to both, because the tonic activity goes down here relative to here, and here it goes up relative to here, so this is excitatory, and this is inhibitory. Okay? Here I drew this little circle here was the EBN. EBN. Boy, what am I going to do now? Okay, so now fortunately I can dump these things and now find. It's so blinding here I can't see anymore. Can you, I can't see anymore, I'm completely blind. Can you, is there a black one in there? No. What's this one here? Right, no, this one, no, this one empty is that. Okay. Huh? As if I've been looking at the sun. Okay, so now on my little schema, I'm going to draw the two superior colliculus maps. One is here. But this is not going to go very far. No, this ain't working either. Gil's not exactly good at supplying stuff that works, is it? Hey, colliculus. Okay. Now we're making a saccade to the right. Which colliculus is going to have a burst?
Left.
So this is a map, right? And the position of the burst is at the place on the map which encodes, say, a right 20-degree saccade. So we have a burst here. And this is the so-called fixation zone. So neurons in the fixation zone, what are they going to do? Neurons are sitting here. They actually send a projection like that. Also connected to the pause neurons like this. If this is a fixation cell, call it Fn. What's the discharge of the fixation cell? Silence. It's the fixation cell has the same discharge as the only pause neuron. It fires tonically, it stops during saccade, and it resumes. That, this is our saccade like that. So fixation cell on both sides have that discharge. They pause during saccades. And you could see that pause neurons also pause during saccades. We know that's an inhibitory input, which the inhibition, I'm sorry, it's an excitatory input because the excite the pause neurons. So, and now what we see here is on the map. Now this is a burst, right? And where does the burst project to? EBN. So we have now a projection that goes like this, right into the EBN pool here. Okay. What does this activity of this cell look like? Our saccade is like this. What's the collicular activity look like? Burst. From here. What this cell is doing. Now the burst here and the burst neurons, that burst has a firing frequency which is proportional to velocity. The burst on the collicular map does not encode velocity. It's a spatial code. The burst here just tells the brain, these circuits, that it wants to generate say, a 20-degree horizontal saccade. Then the question arises, once that happens, how does the saccade, say this burst starts the saccade by activating the burst neuron. Question, of course, is how does the saccade stop? One possibility, being debated, unsure, is that this activity moves on the map until it reaches fixation neurons, which excite pause neurons, which inhibit the burst neurons, and the saccade stops. To move this on the map, you need feedback. How the feedback happens, we don't know. We have no idea how that works yet. It's thought that that feedback might come from colliculus. Others suggest that there's no moving mountain on the collicular map, and that the activity here gets suppressed by cerebellar signals. We don't know that. But all we know is that these commands here, is that visible? Yeah, it's more visible than I can tell by looking here because it's so bright here. That this drive signal comes from cortex. Frontal eye field, show you that in the next slides. So that's better. Let me just check again that this makes sense. Yeah, I think I've drawn all the connections. Yeah. So is that any questions on this? Bearing in mind that I can't see.
Does the EVN connect to the IBN?
So the IBN signal comes exactly from, I thought I'd put it in actually, but it comes from another, it's the same relay, so it goes like this. So the EBN here is connected to 3 neurons. One, the abducens nucleus here, driving the pulse. The integrator, which must produce the signal proportional to eye position. And it activates the IBN, which also has this burst-like discharge. And the IBN inhibits the motor neurons, the antagonist motor neurons on this side. Anything missing from here? So I can't, everything seems complete. Kind of think of this as a kind of a push-pull. Activity goes up on one side and down on the other as the saccadic rate. They start equal, and then on the right, activity goes up, the left activity goes down. All this inner neurons. I think I see some vague pattern right here. Sorry.
If the neural integrator on the left,
Sorry?
if the neural integrator on the left
Yeah.
is being inhibited by the IBN,
Yeah, it goes like it goes through. That's correct. So it's going to be, in a sense, the IBN projects and it reduces the activity. And so if you think of the neural integrator, the inhibitory signal reduces the, it's negatively integrated. Yeah.
So there would also be
Okay, let's go back to the little diagram.
like
So so this is a very complicated reaction. There's things I didn't draw, but let me just. Okay, I'll point with this. This is not working. Okay, yeah, you're asking what?
I was just clarifying.
That's right. There's an EBN. There's just no room. There's an EBN here.
That's what I'm clarifying.
Yeah. There's an IBN here. Yeah, it's bilateral.
Yeah.
There's an IBN here, which also crosses and does the same thing. It goes to the other side. But this IBN here is not active, right? The IBN here is actually.
Oh, I.
Yeah. So the IBN here is inhibiting this because you need this pulse here, right? You need to inhibit because the eye's moving in the opposite direction to this muscle here.
Right.
So you need to hear. And so, but the IBN that's sitting on this side,
Right?
right? But you need an EBN here on this side here, but the EBN here on this side it is not pulsing.
Right.
Right? So that's the reason the IBN would be silent here.
Got it. Thank you.
Okay.
Wouldn't there also be like a connection from the frontal eye field to the excitatory first neuron as well? Direct?
Yes, that's a good question, and that's definitely been shown that there are inputs from the frontal eye field into the brainstem motor circuitry. That's why if you get rid of the colliculus, you can still make saccades, voluntary saccades that come from descending frontal cortical signal. That's true. That's true. And whether they project, now the organization of the frontal projections, I point here, but it's not where I should, the frontal projections into here and the ones that go into here, because as you point out, quite rightly, the frontal eye field also projects directly into the circuitry here. How these connections relate to to the projections from here is not well understood yet. We don't know. You might ask, for example, for a given saccade, we know that this site is active on the collicular map. In the frontal eye field, are they projecting to exactly the same neurons or not? We don't know. And besides, they're interneurons too, because the colliculus also projects to the frontal eye field via interneurons through the thalamus. So this is a very simplified diagram. Yeah.
Art is short.
I don't think I understood your point. Are you talking about two different motor neuron types?
Two different.
Motor neurons are never inhibitory. Motor neurons they activate the muscle. So if they're not firing, your muscle's not active. But the motor neurons they never inhibit the muscle.
My question.
Say again, sir.
The motor.
The motor neurons and interneurons where?
In what?
In the motor neuron pool?
In the.
Abducens. Yeah, that's right. The motor neurons and the interneurons in the abducens have similar discharge. Because there are this is the motor nucleus of the abducens here. And there are two types of neurons. One is the interneuron going here, and the other is the motor neuron going here. These two types of neurons get this burst neuron input onto them. So they have a similar input. And you might call it a pure relay signal. Okay. I say this, but I also always want to warn you, this is the simple version when you're moving the two eyes together. But it happens that you don't move your two eyes together. For example, if you have a target that's going like that and moves towards you like that, say. Okay? So say the target's going here, it goes zoom like that. So what you're doing, if you're looking straight ahead, the two eyes are making saccades but at different amplitudes. Because then the target changes its position relative to your head. So then what you get are disconjugate movements. The two eyes are not making saccades of the same amplitude. And so that requires a more complicated circuitry, which involves this too, because then you need different signals on this neuron and this neuron. Because the eyes are not making the same saccade. Okay? Another example is say the target is moving directly onto this eye like that. So this eye doesn't have to move. But this eye needs to make a saccade to the target here. If this eye is looking straight ahead. So you have saccade in only one eye. So then you get complications in the circuitry, which obviously I can't go into here, right? I can hardly draw this. It would get legible. So, you understand what I mean? Okay. Where are you? Yeah.
So, is this
Yes.
and the neural integrated?
Yeah.
So it's
That's correct.
then why is it called
But if you put a pulse in an integrator, you get a tonic signal out. So an integrator, by definition, just counts. It just counts the spikes. So you have the burst here, which produces the pulse here. But remember that burst, the firing frequency is proportional to velocity. So if you integrate velocity, you get position. But position is a tonic signal. So you take an integrator circuit and you just feed a pulse into it, and the output is a tonic signal, which is the integral of the pulse. That's the definition of an integrator. Integrator is a counter. It counts. So you put in 10 spikes, it might just say counts. It'll count 1, 2, 3. It'll ramp up as the spikes, and after 10 spikes, there's no more input, but the integrator holds its activity. That's one of the assumptions. An integrator must hold the activity. You put a pulse in, and you get a steady state out.
How would that be?
Well, if you don't have an integrator, all you do is put a pulse into the nucleus, but there's nothing to hold the eye. So as soon as the pulse is over, the eye will swing over with the pulse, but then it'll just come back to center. You need a tonic activity to hold the eye in position, because remember that these muscles are like springs too. So if you pull on one side, this gets extended. Even though you actually null the activity here, which you would do because this goes to 0, as you see here, it doesn't mean that you're not pulling this muscle. Muscle is inserted in the eyeball, and it's also inserted in the orbit. So as soon as if you pull it, it's like an elastic band. It wants to bring back the eye to center. So that's why you need tonic activity in this motor neuron to hold the eye off center. Are we through this? So we close this and move on? Yes? Because if you're nodding, I can't even see you.
Your voice is not gonna be recorded.
Sorry?
Your voice is not gonna be recorded.
Yeah, well, because this is not included in the slide package. This is just to be sure that you get to see the circuit and understand it a little bit first, okay? Boy, sometimes I can't believe things work. Okay, so we've gone into this in more detail, but the essence here is that my voice was recorded when I described this, and I've just added knowledge about this business here. You know, how move, stop, how you go from here, how the integrator, how these signals are built up, the burst neurons. In this circuit here, they're all sitting here, and I just made that circuit a little more visible. If I can, is that clear? But it's next interesting exercise for you to work it out. Okay, now let's move, because we're going to move now to cortical control. And this is not going to last terribly long. So I had this slide at the very beginning. Oops, to be recorded. I have to talk here. So we showed, just to review a little bit, that you have the superior colliculus which now you know a lot more about, and the superior colliculus is the common pathway from all these descending command signals from cortex. And there's also the basal ganglia which are involved, which project to colliculus too. So you have the only way you can get cortical control on eye movements is to have the frontal eye field signal, but which bypasses the colliculus directly into the pons here. But if you remove colliculus and the frontal eye field, monkeys cannot make saccades. They're done. Okay. So now we'll consider some details of cortical control. And so there have been many lesions of cortex and subcortex to try to, typically, as you know, see if you can get a link between a lesion and a function. So this is a summary of some of the stuff that's been found. If you have bilateral excision of the frontal left field and the superior colliculus, that abolishes saccades. I've told you that. In fact, I said it twice, I think. Lesions of area LIP, lateral interparietal sulcus, impair visually guided saccades primarily. And if I can go back to say that, if you go back here, you can see that LIP here, whoops, come here. LIP is really in the visual stream. Doesn't like for some reason, I don't know why this is. It doesn't, and so if you take this out, you impair mainly visual guided movements. Lesions of either the frontal left field or colliculus with LIP intact spare visually driven saccades, but not cognitively driven saccades, which would come, say, for example, if you think, oh, I'm going to look right, and you just look right, but that's you're driving that from your intent, not from a sensory stimulus. Lesions of frontal left field with LIP and SC intact impair non-sensory, like memory-guided saccades primarily, for example. And frontal lesions impair intelligent scanning. So that, I thought you'd find that amusing, because there was some very interesting work done many, many years ago in the 50s by a Russian neuropsychologist called Luria, working with a specialist in recording eye movements. And they brought patients into a gallery, an art gallery in Moscow, which is the what's it called? I wrote it down here. The Tretyakov Gallery. Anyone been to Moscow? Seen this? And this painting is by Ilya Repin, who is, he painted this in 1888, 1889. Very large painting which hangs in this Tretyakov gallery in Moscow. And it's worth going on the web to see some of Repin's paintings. They're absolutely incredible. This guy was a great painter, but he was realism. But he was also, this is a little bit of background on this painting. He was on counter to the Tsar. He was pro-revolution. And many of his paintings contained, you might call them anti-government, anti-Tsarist intent or propaganda, such as, for example, this painting people have said that this showed a very positive view of the people for a political refugee that was coming home. And the painting is called The Unexpected Visitor. So that's a bit of the background, but there are many other Repin paintings which are worth looking at. So what Luria and Yarbus was his colleague that was specialized in recording eye movements. Remember, we're a long time ago, and the instrumentation was actually very rudimentary then. But what they did, they brought patients into these, in front of these paintings, frontal patients and normal subjects, and they asked the patients different questions. And it's kind of interesting because you can see that the normal had actually organized scanning patterns, and the patients was completely disorganized. These are very large frontal lesions. And so typically this was what you see in a normal subject as they free view this scene here, which is this painting here I showed you, and they're just free viewing it. Here they ask the normal subjects and the patients. They asked what were the age of the subjects in the painting. Here they asked what the clothing they were wearing to describe the clothing. And here they asked, how long did you think the visitor had been away? So there's intense scanning of the faces of the family that as this unexpected visitor arrives from his political exclusion. So, and you can see that there is very non-specific scanning on the types of the frontal. So this is very famous, highly cited studies, but it's obviously very, it's kind of rudimentary in terms of input-output examination. So the anti-saccade task is another task that's been used to look at frontal lobe function. And what the substance of the task is the following. The subject or the patient and the control subjects initially look at a fixation point which is extinguished here. As time moves this direction, there's a cue that is flashed very briefly here for 100 milliseconds, and then there's period of dark. And the subject is told not to look here, but look to the mirror location of this cue. Suppress a glance here and look here. Suppressing a glance to a suddenly appearing target is not that easy, actually. And then when they if they're successful in making the saccade from here to here, we call that anti, because it's anti to this cue. If they get onto this location successfully, then they have to identify the symbol that appears here. Now, frontal subjects, many of them make 100% mistake on this task. Many do. For example, a normal subject here, this is first a normal subject, would look directly from here, the fixation point, to the symbol and not look at the cue. So in time axes, here the fixation point goes off, the cue is flashed, and then the symbol ultimately appears. And their job is to make the saccade before that appears so that you can get there before it disappears. So a normal subject will make the saccade in this period here, right here, and be able to identify the symbol. Frontal patients, very many, what they do in this task, they actually look at the cue. They can't suppress a reflexive glance to the cue, as if they can't get onto their collicular map and suppress the motor signal on the collicular map. They can't do it. So the result is shown here, that as soon as the cue appears, then they look at the cue, and then they hold there, and then when the symbol comes on, they look at the symbol, because it's the opposite, right? The symbol is here. So they look here, and then they look here when it comes on. And like I said, some frontal patients make 100% mistakes on this. They cannot suppress a reflexive movement. This is what would happen on the collicular map in that context. So bringing together now what we've talked about in the earlier part of the talk with what goes on now in these higher level control problems. So here, this is a normal subject. Fixation point goes off, and then you have the cue is briefly shown. They're not supposed to look at the cue. They're supposed to look to the opposite direction, but before this goes on, as shown here. You get here before this comes on. Now, what happens on the collicular map during this period? During the fixation period, you'd expect these fixation cells to be active. This is a schema, not just to get the concepts, okay? Now, what happens is that the cue comes on here, and that cue would normally require if you looked at it, would require a right saccade, which means you have activity appears in the left colliculus. But the voluntary activity, which says you don't look here, you go here, must be driven by the right colliculus. So you would expect to see, and that's been shown in the collicular map, in monkeys that have been trained to make anti-saccades, you could see that you have voluntary activity starts to appear here, driven by the frontal lobes, and the visual activity here, say, driven by circuits from the visual cortex and parietal cortex, appears here. Now, the job of the system is to get this activity to disappear. So this activity has to be strong enough to suppress this activity via intracollicular inhibitory mechanism, but also intracortical inhibitory mechanism. So the idea is that you need to suppress this activity, and true enough, in normal subjects, this happens. This activity, motor activity, wins, and the subject goes from here to here. Now, in a patient, what seems to happen is that you know that they first look at the cue, they're not supposed to, and then they wait for the symbol to come on, and they make a symbol-driven saccade. The frontal patient, you'd expect this fixation activity to be there, and then the cue-driven activity would appear, but this patient is incapable of driving the voluntary activity, which is going to make the eye go from here to the symbol. So nothing appears here. So that this activity wins over in this struggle between the two sides, and so gradually this activity drives to be beyond the threshold for motor signal. The fixation activity is inhibited, and you get a saccade to the cue, which is not correct, which is not allowed. So the frontal patients make a mistake in this, and then they will only receive, then they only will look at the symbol once it flashes, which then will get activity here. Again, visually driven activity. The cognitive activity, or what you might call the frontal cortical activity, simply does not appear. Frontal eye field lesions also impair memory-guided saccades. So you have neurons in the frontal eye field that have visual responses but no motor-related discharges. Other neurons have motor-related discharge but no visual discharges. And still other neurons have both motor and visually related discharges. Again, to tell you that you have very complicated discharge patterns in these cortical areas. And you also, many of these discharges resemble what you have in the brainstem. So you have the same signal, which you find in many, many different areas. And so this, for example, shows what happens in memory-guided saccades if you put mucimol into the frontal eye field. Now, a memory-guided saccade, I should tell you, is, if you look down here, what you do, the monkey, say, these are experiments done in monkeys. So you have a fixation point that's here. And then while this, I can't do this simultaneously, but say this stays on, and then in periphery there's a target that just flashes like that while this is on. It's just flashed here and it's on. The animal is supposed to not look here, but just keep fixating here. And then when this is turned off, then the animal must make a saccade from here to where the previous target appeared but had been extinguished. That's why it's called a memory-guided saccade. Now, if you deactivate the frontal eye field, like shown here, let's just consider this part of the curve, this graph here. If you put mucimol simply is an agonist of GABA, so you're actually suppressing the activity in the frontal eye field. You could see that in mucimol-injected monkey, they get very, very weak at making contralateral saccades, memory-guided saccades. In fact, you knock them out altogether, whereas they can still make vertical saccades, but they're definitely not as good as a control monkey who is able to look anywhere. Without, of course, as a control monkey, there's no injection. So you have these frontal areas which are involved in more complex saccade tasks, is the best way of putting it. So I think I'll just end here, because I'm sure you've got enough, and me too. And so I could take perhaps questions if I can now turn the lights on, which I don't know how to do. Does anyone know how to do this? Light control? Ah, here we go. Did it. Now I can see you. Questions? No questions? Okay.
I was gonna
Yeah.
um, in the slide you just showed with the frontal eye field, you said that the contralateral side was the contralateral saccade was being controlled in the cortical area. So does that mean that the right frontal eye field controls left saccade?
Yes. Yeah, that's right. Same as colliculus. And there are ipsilateral connections between colliculus and frontal eye field. Hmm? Yeah. Sorry? Speak. What crosses the midline? I mean, the frontal projections? They tend to be ipsilateral because the right frontal eye field controls left saccades. The right colliculus controls left saccades also. So it's a crossed control. Okay.